Welcome to our quick summary series. This is a quick fire summary video. For a longer, more detailed explanation, head over to reviseitright.com. Make sure you subscribe to our channel and turn on that notification bell so you don't miss our 10 minutes a day daily revision, uploaded every day. And if you do enjoy this video, please give it a like. Food is made up of carbohydrates, fats, and protein. These are too large to move into the blood, so must be broken down. This is the function of the digestive system. To aid in this digestion, our digestive system contains enzymes. Enzymes act as a biological catalyst. A catalyst is a substance which increases the speed of a reaction without being changed or used up in the reaction. Enzymes are all large proteins. Reactions involve either substances being split up or joined together. Enzymes help this. Every enzyme has an active site with a unique shape. Here is the active site. The importance in its unique shape is that only the substrate involved in the reaction fits into it. We call this substance a substrate. The theory of how enzymes work is called the lock and key theory. It is called this because the substrate fits into the active site like a lock fits into a key. You need to be able to describe this. You have the active site and the substrate. As said before, these fit into each other perfectly. The substrate fits into the active site of the enzyme, as you can see here. The enzyme breaks down but can also build up the substrate, as seen here. The substrate no longer fits into the active site, so it diffuses away. The enzyme remains unchanged and is now ready to catalyze the next reaction. Just like when we learnt about diffusion, different factors affect how quickly an enzyme can work. The two factors we are going to learn about are temperature and pH. The first is temperature, and here I'm going to draw a graph to show how the rate of reaction is affected by temperature. Look at the graph that I've drawn. What happens as you initially increase the temperature? As we can see here, the rate of reaction increases as we increase the temperature. This is because enzymes and substrate have more energy, so more random collisions between them happen. This increases the rate of reaction. This then reaches an optimum. This is where the enzyme is most active. That's here in the graph. What then happens after optimum? The rate decreases, it massively decreases. This is because the temperature gets too hot, so the bonds within the enzyme break. This changes the enzyme's shape. The enzyme is said to be denatured. Reactions stop taking place. The substrate no longer fits into the active site like a lock and key. You also have to know how pH affects enzyme activity. So I'm drawing a graph for that now. Again, all enzymes have an optimum pH. This could be 8, but could be lower or higher. Here is the optimum. Any slight deviation from the pH causes a reduction in the rate of activity. You see here, if we change the pH by 1, it decreases. And the further you go away from the optimum, the lower the rate of activity. Again, this is because if the pH is not optimum, it breaks the bonds within the enzyme. You have now learnt what enzymes are, how they work, and how temperature and pH affects them. You will find a summary of this information below. Make sure you test yourself using the quiz on reviseitright.com. Remember to subscribe to our channel and turn on that notification bell so you don't miss our 10 minutes a day daily revision uploaded every day. And if you did enjoy this video, please give it a like. We are super excited here on reviseitright.com about the launch of our brand new website. I just wanted to quickly take you through some of the amazing features on our site. Here you can make a profile just like you can on other social media websites, but this one is all about revision, the very first of its kind. You can write in questions here, for example, so can someone help me with the function of bile? You can post it and one of our tutors or another member of the community will get back to you. We're really excited about the revision community we can make here. If you go to the Learn part, here you can have access to text, videos, and quizzes from all the topics you will need in GCSE Science. You'll have access to all the summary videos that we have on YouTube, but also much longer videos that aren't available on YouTube that go into real detail on each topic. We will also have daily and weekly revision challenges for each biology, chemistry, and physics. New challenges each day. And this will link in with the leaderboard so you can compare how you're doing to how everyone else is doing. And really excitingly, you can also book extremely cheap group and private revision classes with one of our tutors. We'll be updating the website all the time, so head on over to reviseitright.com and sign up for a free account today.